Hello, welcome to Lecture 12 of NCBS Neuro, and today we're going to talk about synapse subtypes. I should reiterate that a lot of this is going to be done in more detail by my colleague Shona Chatterjee when he does the next part of the course, but I thought I would give you an overview of the key synaptic types. So, as an overview, we're going to cover simple synapses, which you'll see are not so simple. Then we look at what happens when you put these simple synapses in different places, different targets in different parts of the cell or axon. We look at supersynapses, and then we get into some of the interesting and sometimes simply bizarre cases like dendrodendritic synapses, axonic synapses, gap junctions, completely weird ones like the Martner cell synapse, and broadcast synapses. Why don't we get started? Here's a sort of a general point to be that's useful to keep in mind. It's a rule of thumb. It's not strictly true all the time, but it's a pretty good approximation. It's called Dale's law. And what Dale said was that a cell usually has only one set of transmitters at all of its outputs. However, this doesn't mean that the cell is going to be excitatory wherever it is, because the identity, the nature, the sign of the signal is determined not by the neurotransmitter, but by the receptor that picks up the neurotransmitter and then instructs the cell what to do with it. So, for example, acetylcholine can be excitatory or inhibitory depending on which kinds of receptors take care of it, whether they're the ligand-gated kind or the metabotropic kind, the second messenger type. So you can have different kinds of responses even to the same neurotransmitter. Okay, so let's move on. We'll start with the simple synapse, which is, as I said, not entirely simple. It's got a presynaptic side and a postsynaptic side. But actually, all synapses have a third partner, or almost all synapses have a third partner, and that's the astrocyte, because the synapse is not sitting there in a fluid or in a vacuum. It is sitting there in a complex assembly of many, many other cells, and one of the prominent ones are astrocytes, and they do a great deal of things. They they handle uptake and modulation of the neurotransmitter itself, but they have their own receptors, and they can influence what's happening pre- and postsynaptically as well. So just to get a little bit more detail in what's happening in this so-called simple synapse, the presynaptic side, as you're familiar with, it emits neurotransmitter when the action potential comes in, and we're not going to go into the huge complexity of that. This neurotransmitter may bind to receptors on the presynaptic side itself. It will, of course, get reuptake and it will be degraded and all of that. But on the postsynaptic side, again, there's a lot of things that can happen to it. It can, A, hit a huge range of different kinds of receptors, which, as I indicated, can be different ligand-gated receptors or metabotropic receptors or some combination of them. In many cases, when it hits these receptors, it causes internalization, or it may cause some kind of binding or docking. It may cause trafficking. So there's a lot of interesting things that happen, even in a quote-unquote simple synapse. Again, anticipating what Shona will talk about later in the course, you can have a lot of plasticity at such not really simple, but simple synapses, where you can have postsynaptic plasticity. This is what we generally think of commonly, but it's in which you have the ability of the postsynaptic side to respond to the same signal is modulated up or down. And there's a lot of complicated stuff here, which I won't get into. But you can also have a lot of presynaptic plasticity, which changes, for example, how much neurotransmitter is released and other aspects of the dynamics of the process. So you have a range of things that can happen and a range of receptors, and in fact even a range of partners in what is supposedly a simple synapse. So just bear that in mind. Just to reiterate the point, the same quote-unquote simple synapse can have completely different kinds of responses depending on the input strength. So here we have three cases. This is a review by colleagues of Graham Collingridge. You can have three different kinds of synaptic release probabilities, that is the P bracket R. So let's start with the low release probability, in which case you have only a small amount of neurotransmitter being released. That's the cone of reddish signal here, that's the glutamate. If that happens, then the neurotransmitter only reaches the AMPA receptors. Actually, it will also reach the NMDA receptors, but we won't go into that just yet. And so it'll trigger a certain response based on their properties. If you have a somewhat stronger release probability, then it extends a bit further. It hits more of these receptors. It doesn't really trigger much activity yet in these in the metabotropic receptors, in the second messenger-gated receptors, but you get a different response. And if you have a strong synaptic release, a lot of neurotransmitters coming out, high probability of release, then these are strongly activated, the metabotropic receptors. These are metabotropic glutamate receptors of a certain class. And they will internalize, they'll trigger different kinds of signaling, and quite different things will happen. So, same synapse, same simple synapse, and yet different things are happening depending on the release probability, which can be modulated, again, in complicated ways that we will discuss, or Shona will discuss later in the course. So this was just to reiterate the complexity of even simple synapses. Now let's go to somewhat more interesting cases, or even more interesting cases, if you like. So the kinds that you're familiar with are, you have an axon from the presynaptic cell, and this axon goes and hits a dendritic spine. Okay, that's a fairly typical kind of synapse, and you've seen a lot of that. The schematic, in fact, over here sort of assumes that the synapse is being made on a dendritic spine, which is this little mushroom-shaped structure where the receptors sit. But that's not the only thing that can happen. It can synapse directly on the dendrite, and that will have a different set of properties, because when you have a dendritic spine, it's more like an isolated signaling compartment. When it's on the dendrite, it's not so isolated. The signals travel in a different way, the chemical signals. You can have synapses going on to the soma, and we'll see a little bit more about those. And you can have what are called en passant synapses, which means that it isn't like there's a specific branch of the axon going off to make contact. It's just released along the length of the axon. There's a release site, and that goes and activates, in this case, a dendritic spine, or it could activate something else. Right, so these are all still regular simple synapses, but even here you have this variety of possibilities of location and, in fact, how the junction, how the presynaptic bouton is placed on the axon. It can be at a terminal, or it can be along the length of it. All right, what else can we see? This is what I call supersynapses, or overriding synapses. So here you have your presynaptic cell, and the idea is that this one synaptic input can make all the difference. It can either, in some cases, force the postsynaptic cell to fire, regardless of what else the rest of the cell was doing, or it can completely shut down the postsynaptic cell. For example, as we'll see in a moment, there are synapses which are extremely strong, extremely effective in causing the cell to fire, because they have a huge number of release points. And so it doesn't matter what's happening to all the 100,000 other inputs to the Purkinje neuron, the cell will fire. Conversely, you can have, not necessarily on the same cell, not on the same presynaptic cell, but you could have synapses which come and give a strong inhibitory input to the axon hillock, to the very point at which the axon potential is initiated in the postsynaptic cell. So here, again, it does not matter what the rest of the cell is doing. It does not matter what its inputs are. This cell is not going to fire because it's receiving inhibition right at the point where the action potential would normally have been triggered. Let's have a little bit of a closer look. The climbing fiber input to the Purkinje neuron works like this. You have your climbing fiber come in, and you can see all of the contact points illuminated in yellow over here. Yeah, all around the cell. So it's enveloped the postsynaptic neuron body and proximal dendrites, and this is all from a single fiber. And so you can imagine that once this input comes in, this postsynaptic cell is going to fire, no matter what else is happening in all of its lovely dendritic branches. So next, let's look at the calyx of Hell synapse. This is another supersynapse. It is present in the auditory pathway, some way down, several steps from the hair cell, which goes to VCN, MNTB, which are different steps in the auditory pathway, and finally it comes to the calyx of Held. This is a sort of zoomed in schematic of it, where you have like a clutching glove around a target the synapse is just completely wrapping around the postsynaptic side. And this is a schematic of that, a microscopic view of it, where you have in green, you have the region where the presynaptic side, and the red is the postsynaptic side, and you can see it's just completely enveloped the postsynaptic neuron. And here's a cross section through the same thing. Yeah, so you can see how strong this synapse is going to be. It's definitely going to make this fire. This is, I think, of the order of a micron. Here's another kind of synapses. This is a dendrodendritic synapse. So you're used to thinking of synapses as between an axon on the presynaptic side and a dendrite on the postsynaptic side. Well, here both sides are dendrites. And this is actually quite common in the brain. It's really one of the predominant synapse types in the olfactory bulb, which is what I used to study in the olfactory pathway. But it's there to some extent in many other parts of the brain. It's very prevalent also in the retina, but it's a small fraction of synapses throughout the brain are of this kind. Particularly, as I said, in the olfactory bulb, there's a large fraction there. So let's see what this particular dendritic synapse looks like, and then I'll show you what the circuit is about. So it's not really postsynaptic, presynaptic, but let's think of this blue cell is this huge, big mitral cell over here. These are lovely big neurons. And this is an excitatory neuron in that it uses the neurotransmitter glutamate. And when the glutamate is released, it activates the postsynaptic cell. In this case, let's say it's a granule cell. Okay, so far so good. The presynaptic cell has activated the postsynaptic cell, even if it has done so by its dendrite. You can sort of get used to that idea. But what makes this even more interesting is that the very same terminal of the granule cell now releases GABA, which is an inhibitory neurotransmitter, and it shuts off the very same mitral cell that was giving it excitatory input. So it's kind of an ungrateful situation, right? You get excitation from here, and the immediate response is you shut it up. Okay, so this is present, interestingly, among a lot of the granule cells. So the granule cells have these reciprocal excitation inhibition contacts with the mitral cell, and they don't even bother to have axons. They are axonless neurons. All of their contacts are of this dendrodendritic kind for the granule cells. The mitral cells have an axon which goes off to olfactory cortex, but its dendrites participate in this dendrodendritic interaction. And interestingly, the same mitral cells also have dendrodendritic interactions with another class of cells called periglomerular cells. They're cells which sit around these little spherical regions. So, there are cells which sit around these spherical glomerular regions, which are about 100 microns across, and there's a whole bunch of periglomerular cells sitting around them. And these have exactly the same kind of connection. That is, the mitral cell dendrite excites the PG cells, and the PG cells, ungrateful wretches that they are, inhibit the mitral cells. So this is, as I said, a very common circuit motif, especially in the olfactory bulb. So that was dendrodendritic synapses. What else has nature come up with? Well, if you had dendritic, maybe you shouldn't be surprised that you have axo-axonic. And so let's say here's your standard naive presynaptic cell with an axon trundling along. And you can have axo-axonic synapses where a synapse from this cell, typically an inhibitory one, comes and shuts off the action potential propagation along the axon. That's kind of a general shut off. In other words, it will shut down any and all outputs of this axon that are downstream of this synapse. What's perhaps more common, but far more specific, is if you have a synapse which inhibits, right on the presynaptic side, inhibits the contact to the next cell. So here's your synapse for the presynaptic cell connecting to the postsynaptic cell, the regular synapse. And now you have an inhibitory synapse coming and blocking that one synapse. So this is an extremely precise way of shutting off just one particular synaptic contact without affecting any of the other connections that this presynaptic cell may have made. So there you have axo-axonic. Let's see what else nature has come up with. Well, this is something you are probably familiar with. Again, like dendrodendritic contacts, gap junctions are actually quite common in many regions of the brain, even perhaps more so than dendrodendritic synapses. There are two kinds of gap junctions. Well, there are many kinds, but electrically speaking, two main classes. One is like a resistor. So it's basically like a contact between two cells, between the dendrites or the somas of two cells. And you can represent it as a resistor. It turns out that it's actually a variable resistor. We'll come to that in a moment. Another kind is even more interesting, which is like a diode. In other words, it only lets current flow in one direction. And that's because the gap junctions themselves can have some level of voltage sensitivity. And so they may be such that they will only work, they will only open if the potential on this side is greater than the potential on that side. So current will flow that way, but if this is at a higher potential, current will not flow the other way. So it's acting like a diode. So let's see a little bit about how these work. So gap junctions are hexameric, dual hexameric proteins. That is, they have a hexamer on one side and they have a hexamer on the other side. And these are called connexons, and they need to dock to each other. They need to match, to abut each other in order for the whole connection to be made across the gap junction. Yeah. And they typically come in a fairly large number of them, all closely opposed to each other, as in this schematic. Yeah. The normal intracellular space is quite large, but in this region it's very, very close, 2 to 4 nanometers only. And the middle of these forms a pore, and as I said, this can be open or closed, as indicated in this schematic over here. And then, as I said again, the opening and closing can be gated. It can be gated by the potential, and in some cases it can be gated by various chemical signals or post-translational modifications. So even though these can, in some cases, be thought of even as simple resistors, they are also more complex. They are subject to quite a lot of modulation. Interestingly, in some gap junctions, you can get things going through which are bigger than standard ions. And so you can have contact of other kinds of compounds just beyond just the electrical current flow. So these are all various interesting properties of gap junctions. What else do we have? Here we have something really, really bizarre. This is the mouth nerve cell. This is a key part of the escape circuit of the goldfish. Right. And I won't go through all of the complexities of this little circuit over here. Suffice it to say that this is a very peculiar circuit because it's designed for the utmost possible speed. And one thing which regular ligand-gated channels are somewhat deficient in is that they are not quite as fast. I mean, they're very fast, they're in the millisecond range, but they're not quite as fast as pure electrical propagation. So for example, if you wanted to pass a signal very fast, you might want to use a gap junction. But supposing you want to do something a little bit more complicated, right? So you can imagine that if you had a gap junction, you could pass your excitatory signal if the presynaptic side was excitatory. Yeah? Or if the other side was inhibitory, you could pass the inhibitory signal. But how could you possibly imagine inverting the signal, which you can do very easily if you have a ligand-gated channel? So, surprisingly, nature has come up with a way of having an inverted signal coming through an electrical contact, and this is how it works. So this is the synapse we're talking about, number 4 over here, where you have a nerve which regulates the transmission of information down this axon, and this is how it works. So here is your PHP cell axon. That's the one over here. It comes along, and there's a small region where you have the bare axon, not myelinated. And so, of course, when the action potential comes in, your sodium channels open and huge numbers of sodium ions come in. So far, so good. Then the current naturally flows along the axon, and at the end of the axon, it flows out. Right? And now here what they have is in these mouth nerve cells, in this synapse, they have a tight junction such that the current does not easily flow out, but instead it flows into the next cell. Yeah? And it's the flow of current is substantially through this cap, but also, as I said, around over here. And as we discussed in a previous class, the balance of current flowing through this and through that is such that the mouth nerve cell is actually inhibited when this action potential comes over here. So you have this really bizarre thing where an excitatory input is converted purely through this interesting electrical combination of electrical circuits into an inhibitory signal such that this actually is prevented from firing an action potential. It's called a passive hyperpolarizing field potential is what this synapse achieves. Okay, so that's a really weird one. And finally, here's another class of synapses which doesn't even bother to have very precise localization. And there are two variants of this. One is you can have a neurotransmitter just released at some distance from a whole range of targets, and this is typically a strong neuromodulatory kind of signal where the presynaptic side is designed to affect and modulate a large number of targets. So for example, if you want to have increased arousal or increased or decreased arousal, you might have a transmitter sent out which could affect a large number, typically of metabotropic receptors in a wide range in the vicinity of the synapse. In fact, vicinity can be quite a long range, in the order of tens or more of microns. This is one variant of it. So neuromodulators do a lot of this. Another one is the fact that many synapses trigger the release of compounds which have this remarkable property that they can diffuse right through the membrane, through the cell membrane. And so examples of this are arachidonic acid and nitric oxide. So these are interesting molecules because they go right through the cell membrane. And so that means that any reaction that produces, say, nitric oxide over here will cause a signal to propagate through any and all cells, synapses or cell bodies or axons or dendrites, which happen to be nearby, right? So it's not even clear if this is a synapse, but it's certainly a signal that's being sent from one cell to possibly many other cells. And this is being done through a signal, not really a neurotransmitter, but a signal which just goes right through all the cell walls. So this is, again, a very interesting way in which information is conveyed. For example, in many synapses, there's a retrograde transmission of information. In other words, going from the postsynaptic side to the presynaptic side. And some of these signals are carried by these kinds of signals. So to summarize, the response to inputs in real neurons is actually quite complicated. You have a range of inputs. Excitatory and inhibitory are sort of the plain vanilla flavor. And then you have some of the complicated ones like the axo-axonic synapses or the local diffusion one that we just talked about. You have dendritic synapses and gap junctions. And you, of course, have many second messenger triggering synapses. You have the broadcast neurotransmitters, which are typically neuromodulators. And then you have special inputs which might trigger the entire cell or shut down the entire cell. On top of this, of course, you have all of the other computations that we've discussed hitherto in the class, where, for example, ranging from action potentials going up and down dendrites to passive propagation to local calcium transients, there's all sorts of stuff that is happening. So this was to give you a sense that synapses and neurons can together provide a huge range of varieties of inputs and of outputs and of cellular processing. And so far, we've actually only really focused on one kind of cellular processing. And in lectures to come, we'll also bring up the topic of chemical signal processing. All right. So here's what you probably wanted to hear all along. What is your assignment? So we'll discuss this in more detail in the tutorial, but the idea is to suggest first to think about some of these interesting synapse types and suggest where they may play a particular computational role. I don't want a long, complicated essay type response. I just want a couple of lines naming the synapse and explaining what the computational role is and a reason why the synapse is particularly useful for that computational role. The next question relates to the previous lecture, which was on experimental ways of measuring activity in the brain. And it's a challenge to see if you can figure out how to relate fMRI, which is a very coarse but non-invasive way of measuring brain activity. How can you relate it to activity of individual cells? And I want you to think about that. Okay then.